Hi and welcome to today's session. Today we'll be discussing on lysosomes. I hope you will be familiar with this organelle lysosome. This organelle lysosome is a single membrane-bound organelle which is found in the cytoplasm of eukaryotic cells and contain digestive enzymes. And this in animal cells, they have lysosomes, and instead plant cells have vacuoles to perform the same function. Okay, so lysosome is a single membrane bound organelle which is found in eukaryotic cytoplasm and have digestive enzymes inside it, and in animal cells they have lysosome, and the same function is played by vacuoles in case of plant cells. And it was discovered by a Belgian cytologist, Christian de Duve, in 1955, and he actually named this organelle as suicide bags or suicide sacs. They are most abundant in the cells which are related with the enzymatic reactions like the cells of pancreas, kidney, liver and all. So these type of cells have abundant number of lysosomes. And since they do digestive functions, or they have digestive enzymes inside, that means they perform the role of digestion. That is why they are called as suicide bags or suicide sacs. Coming into the structure, lysosomes are surrounded by single membrane that is the same like the plasma membrane, fluid mosaic model like organization is seen, which is similar to the plasma membrane. So you can see in the inlet here, these are the polar head regions, the yellow colored, and the green colored stands for the phospholipid tail regions. So this is the membrane, single layered membrane of lysosome, and inside they have hydrolytic enzymes or digestive enzymes. So the membrane contains highly glycosylated lysosomal associated membrane proteins, the proteins which are associated with the membranes, which we can see here in the blue color and pink color. These are the membranes which are associated with the lysosomal membrane. We can abbreviate it as LAMP. And the lysosomal integral membrane protein, the protein which I have mentioned these type of proteins in the plasma membrane session. So I hope you know what are integral membrane proteins. So the proteins which are integrated in the lysosomal membrane are called lysosomal integral membrane protein. We can abbreviate it as LIMP. So that constitute about 50% of all the lysosomal membrane proteins. And this lamb and limbs form a coat on the inner surface. This is the outer surface and inside surface, the inner surface of the membrane, and these proteins are thought to prevent the membrane, the lysosomal membrane, from the attack of hydrolytic enzymes which are found inside the membrane. So we know the inside of the lysosome is filled with hydrolytic enzymes. So these hydrolytic enzymes can damage the proteins or the membrane itself of itself. So it doesn't happen like that. Why doesn't it happen like that? Because there will be some mechanism to prevent its own membrane from the digestion. So that role is played by LAMP and LIMP. These enzymes prevent the damage or the digestion of lysosomal membrane from the hydrolytic enzymes. Then the lysosomal membrane also has proton pumps that utilize the energy from ATP hydrolysis and pump H+ into the lumen. So we know that there are several membrane proteins here. One of that is proton pump. Pump is also a protein. What is its function is it pumps H plus ion from the surrounding is cytoplasm. It is found in the cytoplasm, right? So from the cytoplasm, these say for example this pink colored one is proton pump. This proton pump pumps the H plus ions from the cytoplasm, that is from here cytoplasm into the inside of or the lumen of the lysosome. And we know as H plus ion concentration increases, the pH will be acidic. So the lysosomal lumen is always acidic in nature. We know that it is made up or it is filled with hydrolytic enzymes or acids. So that how it occurs is with the help of proton pumps. These proton pumps H plus into the lumen with the help of or with the aid of ATP. ATP hydrolysis powers the energy for them. So that is another function. And the primary function of lysosome is degradation of extracellular and intracellular materials. That is its main function. And for this function, the lysosomes are filled with hydrolysis. Different varieties of enzymes are found in lysosome. That is, first one is proteases. Proteases are the enzymes which digest proteins. Lipases are the ones which digest lipids. Amylases are those which digest carbohydrates. Nucleases are the one which digest nucleic acids, and phosphoric acid monoesterases are also found. So these are the different types of enzymes that are found inside the lysosome. And altogether, the different types of enzymes which are found inside the lysosome are collectively called hydrolases. And these hydrolases cleave the substrate by the addition of water molecules. That is why it is called hydrolases. It cleaves or digests the substrate with the help of cleavage of water. That is why it is called as hydrolases. And most of the enzymes are active in the acidic medium. That is why they need to pump H plus ion into the lumen with the help of proton pumps. Hence, they are called as acid hydrolases. So, the enzymes found inside the lysosome are collectively called hydrolases because they cleave the substrate by the addition of a water molecule. That is why called as hydrolases. And these enzymes are functional only in the acidic medium. That is why they are also called as acid hydrolases. And where it is formed, lysosomes are budded off into the cytoplasm by the Golgi apparatus. We have seen the details in the Golgi apparatus session. And the enzymes that are within the lysosome are made in the endoplasmic reticulum, specifically rough endoplasmic reticulum, which are then delivered to Golgi apparatus via transport vesicles, and inside the Golgi apparatus it undergoes all the modifications for the enzymes, and when the enzymes are fully matured they bud off from the trans phase of Golgi apparatus as vesicles with the acid or enzymes within the vesicles. And types of lysosomes, there are two types, primary lysosomes and secondary lysosomes. The freshly formed lysosomes are the small sac-like structure which enclose the enzymes within it, which are synthesized in the rough endoplasmic reticulum, are called primary lysosomes. Since they store enzymes, they are also said to be storage granules. And when these primary lysosome fuses with some endosome or some other vesicles which are meant for degradation, then it is called secondary lysosome. See in this figure, you can see the rough endoplasmic reticulum budded with ribosomes here. Here the enzymes are synthesized, or the acid hydrolases are synthesized, and from here they are budded off and transported through vesicles to the cis phase of Golgi apparatus. In the Golgi apparatus, it undergoes the modifications and alterations, and all the processes are done, and from here from the trans phase of Golgi it buds off as vesicles. These are the vesicles. Say so for example this one vesicle is lysosome. So this lysosome has inside it the acid hydrolases, and this is called primary lysosomes. When this primary lysosome fuses with some other endosome or phagosome, see this food particles, it is being engulfed by the cell. So it is being engulfed, and this process is called endocytosis. And this vesicle which is formed as a result of endocytosis are called endosomes. It contains the food particle. So this vesicle containing food particle fuses with the lysosome. You can see the fusion process here, and the enzymes inside the lysosome are meant to degrade the food particle in this vesicle. You can see the food particle, this much large food particle is being degraded or digested into this small pieces. So this is called secondary lysosome. When it fuses with some other vesicles for digestion, then it is called secondary lysosome. And after digestion, the food particles or the remaining is eliminated out of the cell, and that process we call it as exocytosis. So since the lysosome performs the digestion of food materials or some other unwanted materials, they are also called as digestive vacuoles. So lysosome is also called as digestive vacuoles. Coming on to the functions, it has several functions: heterophagy, programmed cell death, autophagy, autolysis, fertilization, chromosomal damage, and so on. So first one, heterophagy. What is heterophagy? It is the lysosomal digestion of extracellular materials by the process of endocytosis that we have mentioned in the previous figure. We have seen the endocytosis process, and the endosome gets fused with lysosome and digestion occurs. So it can be of different types, phagocytosis or pinocytosis, depending on what is the matter that is being engulfed into the cell. Then next function is autophagy. Autophagy is a normal physiological process in the body that deals with the destruction of cells in the body itself. The organism's its own cells are being degraded as part of normal physiological process. That function is also performed by autophagy. It is to maintain the homeostasis between the normal cells and the inactive cells. Next is autolysis. Autolysis is the process wherein lysosome digests the cells in which it occurs. That is, a cell in which the lysosome occurs, the lysosome digests its own cell. That process is called autolysis. Autolysis, the lysosome digests its own cell, and it occurs during amphibian metamorphosis and insect metamorphosis. Next function is during the fertilization. During fertilization, what happens is the sperm approaches the egg cell, and the acrosome of the sperm head ruptures and releases some enzymes on the surface of the egg. These are the enzymes that degrade the egg membrane and provide the way for the entry of sperm nucleus into the egg. So that is the function played by lysosome during fertilization. Next is chromosomal damage. Due to the presence of DNase enzyme, that is, I have mentioned earlier, it has nuclease enzymes. One of the nuclease is DNase. The enzyme which damage DNA is called DNase enzyme. So lysosome had an ability to attack chromosome and cause chromosomal breakages. These breakages can lead to many other diseases also. So that was all regarding the functions and structures of lysosome. Go through the video. If you have any query, please let me know. Thank you.